afternoon to all the EAPCET aspirants. So yes, we are in our second session with the chapter photosynthesis in higher plants. Yesterday we have looked into light harvesting complexes, okay, light harvesting complexes which are present in the thylakoid lamellae, where you have two light harvesting complexes, okay. This is the thylakoid lamellae. You have two light harvesting complexes situated here, okay. Light harvesting complex 2 and 1, which is addressed as photosystem 2 and photosystem 1. Each photosystem will be having two sets of pigments. One is core pigment and 2 accessory or antenna pigments. Accessory or antenna pigments. Accessory or antenna pigments include carotenoid, xanthophylls and chlorophyll B. Okay, they include chlorophyll B, carotenoids and xanthophylls. Fine, right? And core pigment includes chlorophyll A, whose absorption maxima of photosystem 2 is P680 and the absorption maxima of photosystem 1, core pigment of photosystem 1 is P700. Clear with these two photosystems? And now we will see photophosphorylation, where the energy which is being released or synthesized in photosynthesis is stored in the chemical form, okay. That is the reason the first step, that is light reaction, is also addressed as photochemical phase, okay. Light reaction is also addressed as photochemical phase. Fine, right? Wherein the light which is absorbed in the form of photons is being stored in the form of chemical energy, that is ATP and NADPH. Fine, right? How ATP is being formed? By photophosphorylation, okay. ADP and inorganic phosphate which is present in the stroma will turn out to form ATP. For this, the energy is required, and as it takes place in the presence of sunlight, it is addressed as photophosphorylation. I hope the term is clear to you. Why is it addressed as photochemical phase and how ATP is formed? So here, as ATP phosphorylation is occurring in the presence of light, it is known as photophosphorylation. We have two types of photophosphorylations, okay. Photophosphorylation. Fine, right? We have two types of photophosphorylation. One is cyclic photophosphorylation and second is non-cyclic photophosphorylation, okay. Cyclic photophosphorylation and second will be non-cyclic photophosphorylation. Fine, right? So what is photophosphorylation? In the presence of light, that is photons, ATP is being formed. ATP is being formed in the presence of photons here. ATP formation occurs from ADP and inorganic phosphate. Fine, right? So this is what happens. Cyclic photophosphorylation is operated in bacterial photosynthesis or anoxygenic photosynthesis. Anoxygenic or bacterial photosynthesis. Where photosystem 2 is absent, photosystem 2 and NADP plus is absent. Okay, and there is only photosystem 1 that is present in stroma lamellae. Only photosystem 1 is present, and that too in stroma lamellae. Clear? This is about cyclic photophosphorylation. Only in stroma lamellae only photosystem 1 is present, photosystem 2 is absent, and there is no NADP plus, which is the ultimate electron acceptor in non-cyclic photophosphorylation. And as photosystem 2 is absent, there is no photolysis of water here. No photolysis of water, and only ATP is formed. Okay, only ATP is formed. Now we will look on to non-cyclic photophosphorylation, which is very important. Non-cyclic photophosphorylation occurs in oxygenic photosynthesis and in higher plants. Okay, this occurs in oxygenic photosynthesis where oxygen is liberated out. Photosynthesis, that is in higher plants, where the absorption maxima is, as it has both PS2, photosystem 2 is present and photosystem 1 is situated in thylakoid lamellae. Thylakoid lamellae. Fine, right? And here photolysis of water occurs. Okay, photolysis of water takes place here. Fine. Where does photolysis of water takes place? In the inner thylakoid membrane towards the lumen. Okay, where the locations or the sites of reactions we have discussed yesterday more clearly. And here ultimate electron acceptor in non-cyclic photophosphorylation is NADP plus. NADP plus, which is situated in the stroma, is the ultimate or final electron acceptor. This is ultimate or final electron acceptor. Clear till here? And then here both ATP and NADPH are formed at the end of non-cyclic photophosphorylation. Both ATP and NADPH are formed. Both ATP and NADPH are formed. Clear till here? Any doubt anywhere, ma? This is the clear differentiation between cyclic and non-cyclic photophosphorylation. Now we will look on to the non-cyclic photophosphorylation more clearly, and then we will learn cyclic photophosphorylation. So first it is non-cyclic photophosphorylation. Non-cyclic photophosphorylation. Here we go. The first step, that is absorption of light in the form of photons. First step of light reaction is clear to us. Second is photolysis of water. So now let us assume, okay, let us assume this is the thylakoid membrane and this is thylakoid lumen. Okay, thylakoid lumen, and this is thylakoid membrane, and this region is stroma. Fine, you are clear with the locations? After absorbing the lights and light getting excited in the form of photons, here you see, let this be PS2 and let this be PS1. Fine, right? So here there is an oxygen evolving complex which is situated on the inner thylakoid membrane located towards the lumen. Here first when photons are absorbed and they got excited to P680 at the reaction center where chlorophyll A is present. Okay, at P680 when they are excited, okay, and the first electron acceptor here it will be, first electron acceptor will be pheophytin. This pheophytin is situated on the outer side of the thylakoid membrane. Okay, so this is the first excitation of electrons. When photons are being excited, the electron, first electron is being transferred. First electron acceptor is pheophytin. But the continuous flow of electron transport is not supplemented by photons. Okay, it is supplemented from oxygen evolving complex here, where inside lumen the water molecule, okay, the water which is present inside the thylakoid lumen, this is photolysed to 2 protons plus 2 electrons and oxygen atom. Fine, right? Protons are present inside the thylakoid lumen, whereas electrons, they get excited and the first electron acceptor will be pheophytin. First, it is due to the excitation of photons, electrons will be accepted by the primary electron acceptor, that is pheophytin. But the continuous flow of electrons is supplemented by photolysis of water which is happening inside the thylakoid lumen. Okay. Now, the pheophytin which is situated on the outer side of the thylakoid membrane, this will give the electrons not to an electron acceptor but to a proton acceptor, that is plastoquinone. Okay. Plastoquinone will start quinone cycle here. Soon as it gets the electrons from pheophytin, this will parallelly accept protons, from stroma, and this gets converted to plastohydroquinone, PQH2. This gets converted to PQH2. Fine. And then electrons are being pumped to cytochrome B6F complex, whereas protons are being sent to the lumen. Fine. Now, the ultimate electron acceptor over here will be plastocyanin. Okay, plastocyanin will be the ultimate electron acceptor here. Clear till here, ma? See, in photosystem 2, where P680 is the reaction center, first the electrons are excited to P680 from photons, and the primary electron acceptor will be pheophytin. The electron flow will be continuous because of the photolysis of water which is happening inside the thylakoid lumen. First electrons are given to pheophytin, then quinone cycle will be operated here. Pheophytin will transmit the electrons not to an electron acceptor but to a proton acceptor, where here plastoquinone will take the protons from the stroma and this gets converted to plastohydroquinone. From this PQH2, protons are pumped to the lumen, whereas electrons are given to cytochrome B6F complex, and the last electron acceptor of photosystem 2 is plastocyanin. Okay, plastocyanin is the last electron acceptor of photosystem 2. This is photosystem. Fine, right? Now, in photosystem 1, the electrons whichever are being transferred from plastocyanin, they get excited and the reaction center over here is P700. Fine, right? From here the electrons are being transferred. Again the downhill transport of electrons will take place by ferredoxin and ferrous sulfur complex and ferrous sulfur complex. Fine, right? And here the ultimate electron acceptor is NADP plus. This NADP plus is situated towards the stroma. Okay, NADP plus, which is situated towards the stroma. This accepts the electrons. This accepts the electrons, and this gets converted to NADPH plus H plus by the enzyme NADP reductase. Fine, right? NADP plus is converted to NADPH plus H plus, and the NADP is reduced to NADPH by the enzyme NADP reductase. So here first excitation of electrons to the reaction center in photosystem 2 is an uphill reaction, followed by continuous downhill reaction in the sequence. The electrons you can see, right, this is downhill, and then again uphill to P700, and then downhill flow of electrons till NADP plus, and NADP plus, which is the ultimate electron acceptor, this where the electron it accepts the electrons and reduces to NADPH. NADPH formation takes place inside the stroma, and NADP reductase is the enzyme which is reducing NADP plus to NADPH plus H plus. So at the end of non-cyclic photophosphorylation in this process, you can see NADPH formation, but there is no ATP formation that you have observed till now, right? Now we will see ATP formation. Till here everything is clear, right, ma? Any doubt anywhere, you can drop a message in chat box. Any doubt anywhere, you can ask me. Fine. Now we will look into chemiosmotic hypothesis, which is proposed by Peter Mitchell. Chemiosmotic hypothesis. Here we move on. How ATP is being formed. So there are three main reasons, if you can see, the non-cyclic photophosphorylation. First, due to photolysis of water, protons are being accumulated inside the lumen. That is the first reason. And second, protons whichever are present in the stroma, those are being taken by the quinone cycle, okay, by plastoquinone, and again they are passed on to the lumen. This is the second reason where protons are reduced in the stroma and increased in the lumen. And third reason, if you see, whatever minimal protons are present in the stroma are then utilized by NADP plus, and that is being converted to NADPH plus H plus. So because of the reduction reaction, again protons are being consumed from the stroma. These are three main reasons where proton gradient, protons are being reduced inside the stroma and increased in the lumen, okay. So the main reason for the ATP formation or proton gradient generation is reduction of protons. Reduction of protons in stroma. Here can you see reduction of protons in the stroma. There are three main reasons, if you can observe. First is increase in proton concentration inside the lumen, where pH will be reduced inside the lumen. Fine, right? And next is through quinone cycle, okay, where proton gradient is again increased inside the lumen. Third is due to the reduction step of non-cyclic photophosphorylation. Mention out the reasons carefully. First is photolysis of water increases protons in thylakoid thylakoid lumen. Fine, right. Second is protons are pumped from stroma to lumen by Quenon cycle. Which again increase the proton concentration in thylakoid lumen. Fine. And third reason, if you see, inside stroma where NADP plus is reduced to NADPH plus H plus. Fine, right? This reduction to NADPH is again reducing the proton concentration in stroma. Fine, right? These are the main reasons for increase in proton concentration inside the thylakoid lumen and reduction in proton concentration inside the stroma. Fine, right? So, here you see the main reasons, okay, where pH is reduced inside the lumen and pH is increased inside the stroma. And 4 requirements are there for chemiosmotic hypothesis to operate. First is membrane, that is thylakoid membrane, right. Second is a proton pump. And third is proton gradient. Fourth will be ATPase. Fine. So, here you see, first the proton pump which is operated is F0 F1 pump. Okay, it is F0 F1 pump. Can you see here? F0 is embedded inside the thylakoid membrane and F1 is towards the stroma. Okay, F1 is towards stroma. In one run, 4 protons are being released, and inside stroma, ADP and inorganic phosphate which is there by the enzyme ATPase. ATP is being formed. Fine, right? And this F0 F1 pump is also known as ATP synthase pump. It is from which direction to which direction? From lumen to the stroma. Whereas in mitochondria, if we have to compare once, in mitochondria if you see, in mitochondria, where outer mitochondrial membrane, inner mitochondrial membrane, that is cristae which is present, F0 F1 pump will be from intermembrane space to the mitochondrial matrix. Okay, it will F0 F1 pump will be from intermembrane space to mitochondrial matrix. Here it is from lumen to the stroma. Matrix, stroma is also called as matrix of the chloroplast. Clear till here? Any doubt anywhere about chemiosmotic hypothesis and non-cyclic photophosphorylation? I hope everybody are clear with how NADPH and ATP are formed. Fine. Now, what are the end products of light reaction? Here we go with the end products of light reaction. Include ATP and NADPH, which are the assimilatory power which is formed, and oxygen which is evolved due to photolysis of water. ATP and NADPH are formed inside the stroma whereas oxygen is evolved inside the thylakoid lumen. Fine. Utilizing this assimilatory power which is released in the stroma, glucose formation takes place. Fine. Now, we will look into the biosynthetic pathway, that is biosynthetic pathway or light independent pathway. Okay. Look at here. Biosynthetic phase, or light independent phase. Biosynthetic phase and light independent phase. We have 3 cycles we have observed: C3 cycle, C4 cycle and CAM cycle. C3 cycle is operated in all the plants. C3 cycle is observed in all the plants. C3, C4 and CAM, all the plants C3 cycle is seen. This is given by Melvin Kelvin. After Second World War, when there is huge research going on the radioisotopes, Kelvin has performed very good experiments on algal photosynthesis by synthesizing a radioactive isotope, that is C14. Utilizing C14, he has conducted algal photosynthesis and the model organism is chlorella, where he demonstrated that the first product that is formed after carboxylation, the first stable organic intermediate that is formed after primary carboxylation is 3-phosphoglyceric acid. Okay. This is 3-phosphoglyceric acid, which is a 3-carbon compound. This occurs in all the photosynthetic plants. Okay. 3-PGA, which is the first formed organic intermediate, 3-phosphoglyceric acid, which is 3-carbon compound. That is why it is also known as C3 cycle. How C4 cycle is discovered? This is discovered by Hatch and Slack. So, the name is also given like Hatch and Slack pathway. The name is C4 cycle because here the first stable product formed is, or the first stable intermediate that is formed after primary carboxylation is oxaloacetic acid. Oxaloacetic acid, which is 4-carbon compound. This is being converted to malic acid or aspartic acid. Okay, OAA is converted to malic acid and aspartic acid. This occurs in all the C4 plants, and here there is an adaptation, that is Cran's anatomy, which is observed in dry tropical plants which are in tropical climates. Mostly this C4 cycle occurs in most of the monocots like sorghum, maize and sugarcane. C4 cycle occurs in most of the monocots like sorghum, maize and sugarcane. Only dicot that conducts C4 cycle is amaranthus. New point to be noted. Next is CAM cycle. CAM cycle is also known as crassulacean acid metabolism. Here also the first stable intermediate formed is oxaloacetic acid. This occurs in crassulacean family members, and here you can see CAM cycle operates in usually desert plants or the plants belonging to the family crassulacean family. Okay. Here there is an adaptation that stomata opens during night time and closes during daytime. This is known as scotoactive stomata. Scotoactive stomata. Fine, right. Examples you see, succulent plants include cactus, agave, opuntia and aloe vera. These are the examples of succulent plants. This CAM cycle is also seen in few tropical epiphytes like orchids. Clear, right? Any doubt anywhere? Yes, ma, you can't skip diagram based questions. Though the exam is in online mode, you can't skip diagram based questions because diagram based questions are very important. even though diagram may not be given in your question, but from diagram statement based questions are asked, assertion reason type of questions will be picked from diagrams. So, you cannot skip diagram based questions. The concept which is related to each and every diagram mentioned in your text, you have to go through it. Fine? Done? Yeah. Now, we will clearly look into C3 cycle. C3 cycle is operated in 3 main steps. First is carboxylation. Second, reduction, and third is regeneration. Fine? Carboxylation, reduction and regeneration. Carboxylation, which is the first step, that is fixing of carbon dioxide from atmosphere, this is the most crucial step of C3 cycle. Okay? Carboxylation is the most crucial step of C3 cycle. This carboxylation is done by what enzyme? Rubisco. Rubisco is the most abundant enzyme on the earth. Fine, right? Here there is some points that have to be discussed from Rubisco. Rubisco, as the name itself says, ribulose bisphosphate carboxylase oxygenase, right? Ribulose bisphosphate carboxylase oxygenase. So, it has both the enzymatic roles, that is, it can function as carboxylase and it can function as oxygenase. Carboxylase and oxygenase. Whenever Rubisco is performing carboxylase activity, here carboxylation takes place, where carbon dioxide fixation occurs. Fine. Oxygenase, when Rubisco is performing oxygenase activity, here photorespiration occurs or C2 cycle is operated. C2 cycle or photorespiration. When does Rubisco performs oxygenase activity? Whenever the oxygen concentration is more when compared to carbon dioxide, then Rubisco binds with oxygen and performs oxygenase activity, photorespiration occurs. Photorespiration or C2 cycle occurs only in the C3 plants. This occurs only in C3 plants. Fine. Whereas Rubisco, when along with carbon dioxide, okay, when it performs carboxylase activity, Rubisco along with carbon dioxide, when it fixes carbon dioxide and forms carboxylase activity, carbon dioxide fixation occurs, and here itself you can see the 3 main steps of C3 cycle. Here this operates what cycle? C3 cycle. Fine, right. So, you are clear with the 2 activities of Rubisco. After carboxylation, the first organic intermediate which is formed is 3-phosphoglyceric acid. For 1 carbon dioxide, RUBP is the acceptor, the enzyme that is catalyzing the reaction is Rubisco. But what is the acceptor of carbon dioxide for the first step? Carbon dioxide acceptor is RUBP, that is ribulose bisphosphate, which is 5 carbon compound. Carbon dioxide, which is 1 carbon, RUBP, which is 5 carbon. In the presence of the enzyme Rubisco, this fixes 2 molecules of 3-phosphoglyceric acid, which has 3 carbons. Fine, right? 2 molecules of 3-PGA. Now, reduction step is very huge, which is 7-step long process, but you remember from 3-PGA it gets reduced to 2 molecules of glyceraldehyde 3-phosphate. Okay, 2 molecules of glyceraldehyde 3-phosphate means on total it is having 6 carbon, and for this reduction step for 1 carbon dioxide, the reduction step will consume 2 ATP and 2 NADPH is consumed for the reduction step, and 2 ADP, 2 NADP plus is being released. From the 6 carbons of 2 molecules of glyceraldehyde 3-phosphate, 1 carbon will be leaving each cycle for the formation of glucose, that will be leaving into the stroma, whereas 5 carbons will be regenerated back for the synthesis of RUBP. After carboxylation, it is regeneration step which is most crucial for C3 cycle to continue the cycle uninterruptedly. To continue C3 cycle uninterruptedly, regeneration step is very important. Here 1 ATP is being utilized in this step for 1 carbon dioxide. Clear till here? So 1 carbon dioxide to be fixed, how much amount of energy is required? On total 3 ATP and 2 NADPH. And to form 1 glucose, how many cycles have to happen? 6 cycles, and 6 carbon dioxide molecules have to be fixed, because from 1 cycle only 1 carbon is leaving the chain to form the glucose. Glucose is 6 carbon, so 6 cycles have to happen. Now let us look into the bioenergetics of C3 cycle for the fixation of 1 carbon dioxide and for the fixation or formation of 1 glucose. Yeah. Here we go. For the fixation of 1 carbon dioxide, how much energy is required? We discussed 3 ATP, 2 ATP in reduction step and 1 ATP in regeneration step, and 2 ATP, 2 NADPH in the reduction. For formation of 1 glucose, 6 cycles will be done, 6 carbon dioxide fixations are required. So 6 into 3 ATP is total 18 ATP is required, 6 into 2 on total 12 NADPH are required. Fine, right? How much ATP and NADPH are required to reduce 4 molecules of G3PA? Okay, to reduce 2 molecules of G3PA, 3-phosphoglyceric acid, to reduce 2 molecules of 3-phosphoglyceric acid, we require 2 ATP and 2 NADPH. And for formation of 4 molecules of PGA, phosphoglyceric acid, how much amount of energy is required? 4 ATP, 4 ATP and 4 NADPH. Fine, clear till here? Any doubt anywhere? And then what is the amount of assimilatory power that is required for 1 sucrose formation? For 1 sucrose formation, how much assimilatory power is required? When assimilatory power term is mentioned, remember he is using it both for ATP and NADPH. Fine. For formation of 1 sucrose, it will be double than that of glucose. Glucose it is 18 ATP and 12 NADPH. Sucrose it will be 18 plus 18. 36 ATP. And how much NADPH? 12 plus 12. It is 24 NADPH. Clear? Any doubt anywhere, ma? Clear till here? Yeah, this is all about C3 cycle. Now we look on to C4 cycle. C4 cycle is operated only in dry tropical plants. Dry tropical plants, which are C4 plants. Here you see C4 plants, they have Kranz anatomy of bundle sheath cells. What is Kranz anatomy? Bundle sheath cells will surround along large cells, okay. The cells which are large in size, they surround around the vascular bundles of the cell, okay. Bundle sheath cells, they will be surrounding around the vascular tissue in 2 to 3 rows, in multiple rows, okay. What are the characteristic features of this bundle sheath cell? What are the characteristic features of this cells? Remember, bundle sheath cells consists of large amount of chloroplasts, are present inside the bundle sheath cells. These chloroplasts are agranule. A granule chloroplast. There is no grana inside this chloroplast, only stroma is present. Stroma lamellae is present. Okay, and there is an enzyme, that is Rubisco, which will fix the carbon dioxide for the second time in the bundle sheath cells. Remember this point. Bundle sheath cells will surround as a vrith. Crans means vrith. Okay. Crans means vrith. Okay. Bundle sheath cells, which are large in size, they surround around the vascular tissue, and this bundle sheath cells will contain large amount of A granule chloroplast. Okay, they contain large amount of A granule chloroplast. Here second time carbon dioxide fixation occurs. First in the mesophyll cells. In mesophyll cells, C4 cycle will take place and it has what? Granule chloroplast. And inside bundle sheath cells, C3 cycle will take place, which has A granule chloroplast. Granule and A granule chloroplast. Clear till here? Clear with the anatomy of C4 plants? Any doubt anywhere? If you are clear with C4 cycle, look into the mechanism of C4 cycle. Here in C4 cycle, there is spatial separation of carboxylation reactions to happen. Okay. The spatial separation is in between mesophyll cells and bundle sheath cells. Mesophyll cells and bundle sheath cells, they are separated spatially. Okay. And these cells are connected by plasmodesmatal connections. The atmospheric carbon dioxide is absorbed in the form of hydrogen carbonate and it is fixed by the acceptor PEP, phosphoenol pyruvate. First, carbon dioxide is reduced to hydrogen carbonate by the enzyme carbonic anhydrase, and this PEP, which is the primary acceptor of carbon dioxide in C4 cycle, this gets converted to carbon dioxide, which is 1 carbon, and phosphoenol pyruvate, which is 3 carbon. On total 4 carbon is fixed. First organic intermediate is oxaloacetic acid, which is 4 carbon compound, and the catalyst which is here is pepcase, phosphoenol pyruvate carboxylase. Unlike Rubisco that you have observed in C3 plants, which has both carboxylase and oxygenase activity, here pepcase will perform only carboxylase activity. Only carboxylase activity is observed. Here oxaloacetic acid by the enzyme malate dehydrogenase, this gets converted to malic acid or aspartic acid. Fine, right? Malic acid or aspartic acid is transferred to the bundle sheath cells via plasmodesmatal connections, and once the malic acid or aspartic acid entering into the bundle sheath cells, these are 4 carbon, right, decarboxylation reaction happens, wherein 4 carbon is separated to 3 carbon pyruvate and 1 carbon dioxide. This 1 carbon dioxide Kelvin cycle is operated. Okay, Kelvin cycle is operated inside the bundle sheath cells, wherein the amount of energy that is released is 3 ATP and 2 NADPH is the amount of energy that is released, and the enzyme here is Rubisco. Rubisco is the enzyme. Okay, the energy that is released is 3 ATP and 2 NADPH, and the enzyme is Rubisco. From here, if you see, the pyruvate, whichever 3 carbon is synthesized inside the bundle sheath cells, they are transferred again to the mesophyll cells via plasmodesmatal connections, and for this transfer the enzyme required is pyruvate phosphate dikinase, which synthesize phosphoenol pyruvate, and here how much amount of energy is consumed? 2 ATP. So, if you see the whole cycle, you can see 2 carboxylation reactions that are happening, one in the mesophyll cells and the second is in bundle sheath cells. So, for 1 carbon dioxide which is entering the cycle, how much amount of ATP is required or consumed? 3 ATP in the Kelvin cycle and 2 more ATP for the regeneration of pyruvate, and here 2 NADPH. So, for if you see the bioenergetics of C4 cycle, how much amount of energy is required, ma? For fixation of 1 carbon dioxide. For fixation of 1 carbon dioxide, we require 3 ATP and ATP for Kelvin cycle and 2 ATP for C4 cycle, total 5 ATP are utilized, and 2 NADPH for Kelvin cycle. Fine. For formation of one glucose molecule here, 6 cycles have to happen, where carbon dioxide is fixed for 6 cycles. Okay, 6 cycles have to happen. 5 into 6, total 30 ATP is being synthesized and 12 NADPH is being synthesized. Fine, right? Total 30 ATP and 12 NADPH for the formation of one glucose molecule. Clear till here? And few questionnaire on bioenergetics. You can see here how much extra ATP is required to fix 6 carbon dioxide molecules. To fix 6 carbon dioxide molecules, how much extra ATP is required? To fix 1 carbon dioxide, extra ATP required is 2 ATP. For 6 carbon dioxide, 2 into 6, it will be 12 ATP. Remember, analyzing question is important. Here he didn't ask total ATP for C4 cycle. He just asked extra ATP required for C4 cycle. Now, how much extra ATP is required for the formation of 1 sucrose? For the formation of 1 sucrose, how much extra ATP is required? For 1 glucose formation, it is 12 ATP. For 1 sucrose, it will be 12 plus 12, it is 24 ATP, right? That's it. It is as simple as it is. This is how you have to look into the questions. Next, CAM cycle. Fine. In C4 cycle, if you see, for 2 carboxylations to happen, there is spatial separation, but here 2 carboxylation reactions are separated by time. If you see, in CAM plants, there is an adaptation by stomata, where stomata opens during the night time and closes during daytime, which is known as scotoactive stomata. So, first carboxylation or carbon fixation will happen during night time by the enzyme peptase. Okay, peptase will fix the carbon dioxide during night time. So, first carboxylation or carbon fixation, carbon dioxide fixation occurs during night time. The reactions and energetics, the mechanism and energetics are same as that of C4 cycle, but here they are separated by time, that is it. There they are separated by space. Okay. So, here you see oxaloacetic acid will be converted to malic acid or aspartic acid by the enzyme malate dehydrogenase, and here malic acid decarboxylation takes place. Malic acid is stored in vacuole, and during daytime decarboxylation occurs. Carbon dioxide, whichever is released into the mesophyll cells during daytime by the enzyme Rubisco, it will be fixed again. Kelvin cycle will be operated again during the daytime. Fine, right, by the enzyme Rubisco. Fine. Second carboxylation occurs during daytime, first carboxylation during night time, and here the energetics are completely same as that of C4 cycle. For 1 carbon dioxide fixation, it is 5 ATP and 2 NADPH required. For 1 glucose formation, it will be 30 ATP. For 1 glucose formation, it will be 30 ATP and 12 NADPH. Clear, right? With this, all the biosynthetic reactions are done. We are left over with 2 more concepts from this chapter. One is photorespiration or C2 cycle, which is operated only in C3 plants, and second is factors affecting photosynthesis. These 2 concepts we will be exercising in our next class, followed by EAPCET previous year questions from this chapter, and then we will move on to respiration. Fine, right? So, that is all for today's session, ma. Just go through the concept. If you are having any doubts, please drop messages in comment box. I will be addressing all your doubts.